how the chlorophyll actually takes that light energy and then does something chemically with it. And so these chlorophyll molecules are going to be located, actually embedded into our thylakoid membrane. So we can visualize that like this. So this will be our thylakoid membrane here. And outside of the thylakoid membrane, we have the stroma. So that's the space in between the membrane and the outer edge of the chloroplast. Stroma. And then inside the thylakoid, we've got the lumen. And so our plastids, our chlorophyll, are going to be associated with these protein complexes called photosystems. And we have two photosystems in the chloroplast. And we call those, I'm going to abbreviate here, photosystem 2 and photosystem 1. Now don't let the numbering of the photosystems throw you. They were numbered in the order in which they were discovered. So we'll see that the reactions actually start in photosystem 2 and then go to photosystem 1. You actually run into this quite a bit in science. Odd naming scheme is based on when things were discovered or who discovered them. So anyway, the chlorophyll, along with other pigments, are associated with our photosystems. And so what happens when a photon of light in that visible spectrum comes in and hits one of these pigments is that an electron in the chlorophyll, specifically in this ring here, that electron is going to be excited. Not excited because it's part of an awesome process, but actually chemically excited. It's going to move from its normal orbital up to a higher orbital. And so that's unstable. And so what it's going to do is it's going to actually bump into a neighboring pigment and then transfer the energy from that excited electron to a neighboring pigment. And so the electron in this first pigment will fall down to a stable energy level, and then the electron in the next pigment will get excited. And through this process, the energy from the photons are going to be funneled into what's called the reaction center in the photosystem. And so we'll have our energy is transferred from pigment to the reaction center, at which point it will get to a special chlorophyll A, where instead of simply exciting the electron, that electron will be excited and then actually removed and passed to another molecule. So I have an electron, and that's going to get passed over to an electron acceptor. All right, so now we've removed an electron from our chlorophyll, so we need to replace that. And so we're going to do that by splitting water and removing an electron from water, and that's going to give us, we'll say, half of an oxygen and 2 protons. So this is where we're going to be using water, which we have in our overall equation, and this is also how we're going to be producing our oxygen. So now we have our electron, we've moved it to another protein in the membrane, and now we're actually going to transport that electron through a chain of different proteins in the membrane. And so does that sound familiar? I see some heads nodding. So why does that sound familiar? And where did we see that before? Yeah, and so we're going to be doing basically the same thing that we did in class on Wednesday, in that we're going to be transporting our electron down an electron transport chain. And while we're doing that, we're going to be pumping protons from the stroma into the lumen using the energy from that electron. So protons. All right, so we've now pumped a bunch of protons into the lumen, and our electron has now arrived at photosystem 1. Photosystem 1 has that same array of pigments around it, has different chlorophylls, so it too can absorb energy from light. So we'll have another photon of light that's going to come in and hit the pigments in that photosystem, and it will excite the electron in the reaction center, and that's going to get passed to another molecule. And that electron that we've now gotten rid of here will be replaced by the one that came from the electron transport chain. And now our last step here is to pass the electron off to a final electron acceptor. And in this case, it will be NADP. And we'll convert NADP plus to NADPH. So in our electron transport chain in respiration, we are passing the electrons to NAD and creating NADH. In the chloroplast, we use NADP and convert that to NADPH, basically doing the exact same thing. All right, so now we've added an electron to our electron carrier, and we built up a whole bunch of protons in the lumen. So what do you think we can do with a whole bunch of protons on one side of a membrane?
Sucks through and goes through.
What's that?
Do we have a synthase, another like a similar functional mechanism that uses that power to create? Is it not ATP in this? Is it something else? Or?
It is ATP.
ATP.
Yep. So we're actually going to have ATP synthase, just like we did in the mitochondria. And that ATP synthase is going to be pumping the protons from the lumen back into the stroma. And so it works just like the one in the mitochondria. I think we had our ATP synthase played by Josh last time, so it spins around each time it synthesizes an ATP, which is pretty cool. And so we'll be converting ADP to ATP. So now in the stroma, we've built up a bunch of electron carrier, NADPH, and also a bunch of ATP, which we will use subsequently in the Calvin cycle. All right, so we've been talking for a little while now